Thank you very much again. Our next speaker will be Wouter van den Broek. Sorry for my bad Belgian. He will deliver what I would consider the epitome of an art science talk, because it will be about both. The making of 69 days of close encounters in the science lab.
In the spring of 2009, the Science Gallery in Dublin organized an exhibition, an art science exhibition, on the topic of contagious phenomena and methods of containment. Now, this is a sample of the kind of elements or the kind of exhibits that were shown. It was about clinical contagion, but also about the spreading of ideas as infections. Now, the visitors, when they entered the exhibition, were greeted by these hosts, which, as you can understand, made everybody feel really comfortable. Especially when they started scanning for some unnamed pathogen and started handing out measurements, protective measurements, including lanyards with some undescript electronic device. Now, this electronic device is actually an active RFID badge developed in the context of the SocialPatterns project, which is an international project collaboration between different groups in Italy, France, and Germany that is interested in figuring out fundamental patterns in coordinated human activity, and in particular based on complex networks science. One of the results of this project is a SocialPatterns, is a technological platform, which is sometimes referred to as a SocialPatterns platform, but the SocialPatterns project is much broader than just this platform. But it consists of these devices, the aforementioned devices, which are worn by people and which are used to detect to collect high-resolution data on physical proximity among people. And high-resolution, I mean like 1 meter, 1.5 meters, and 20 seconds time slices. Now, so two people wear these devices, and when they detect each other's proximity, they scream out their presence to the stations which are installed in the room. Now, typically to cover large areas, we use several or multiple of these stations, which all forward the data to a central server, which then either stores the data for later processing or analysis, or we can also do this in real time. So we have an event stream of proximity events, which we can then visualize in real time, which enables us to place within the room in which we deployed this platform to interactively or dynamically show the current social network. But it's not really the social network, it's like it's in something that approximates the social network, because it's just physical proximity. Now, to give you an idea, I'm going to show a little movie which takes a selection from one of the experiments we've done. Now, what you're going to see is the outer circle are representations of the different stations that we had in the different rooms. Notice in particular Sala A, which was a main room in which there was a presentation going on. We had 2 readers there. And then on the left we have 3 readers in the bar, which is a coffee bar. And the slice I'm going to show is just before the end of the first session, where the people are still sitting in a room listening to somebody speaking. and then the session ends. All of a sudden, all these people start interacting with each other. Right, they start facing each other actually. So, but it's a good approximation of interaction. They all move to the bar, have their coffee, have a lot of talking and discussion because the session was really interesting. And then they move all back to the room because then the next session is going to start with the next talk. This, of course, obviously is a fast-forward replay of the actual data. Now we have deployed this system in more than a dozen occasions, ranging from schools, hospitals, conferences, from very scientific objectives, all the way to broader art science deployments. So this is an example of some of the results of a very scientific deployment in a hospital in Rome, where we looked at the different types of roles that the people had that we were looking at. So we had children, parents, nurses, doctors. Or we have something very art science in the Laboral in Gijón, which is an amazing museum. And there we did a workshop looking at, based on Latour actually, the actor network of objects and participants in the workshop. So yeah, fancy movement. Oops, sorry. So, and another of these projects, of these more art science projects, was what we call Infectious Sociopatterns. So it was a collaboration between the SocialPatterns project and then for this infectious exhibition. What we did there is we extended the device with a little bit of functionality. It was just a little bit, but very effective. Namely that each device was either in an infected or a non-infected state. And so everybody got a device which was initially not infected. But by being in the proximity of somebody who was infected, there was a probability that you could get infected. Now there was a little light blinking on the device, and normally it blinks like every couple of seconds just for us to see that the battery is still okay. But when you got infected, it started blinking really fast. So then we also had visualizations in the room, in the exhibition area, where when a node was infected, it was red. When it was not infected, it was green. The people could also see on the badges what their ID was, and they could correlate with, so they could figure out which node represented them on the visualization. We also had, like in certain parts of the exhibitions, alarms that went off when somebody with an infected badge passed by. We had like little healing stations where if you got infected, you could get healed again. So people going in front of the screen just to get healed. And there, for example, in the back you see like a screen. So this was like to introduce a whole dynamic, of course, to like have a real-time experience of what it is to have an infection going on in real time without actually hurting anybody. The funny thing though is on some of the days we consciously or like on purpose did not explain a lot to the people what was actually going on, just to see how they responded to it when they had knowledge or when they did not have knowledge. And the funny thing is the kids always understood it quite rapidly. So once they got the thing, they started running after each other. It's like madness all over the place. So, of course, another thing that I need to note is like this 2 or 3 weeks after this exhibition got started, the swine flu broke out. So all of a sudden there was a lot of attention for this kind of exhibition, and they even blamed us and said, this is a promotion stunt, of course, right? Nothing to do with it. So based on so actually this exhibition went on for 3 months. So 3 months, every visitor that came in, or almost every visitor, because of course we did not force it on people, but almost everybody participated, got a badge. That means in total we had about 30,000 people coming in during those 3 months wearing a badge. So for all these days we measured the interaction patterns between all these people. 30 days, 96 days, 69 days actually, hence the title of the talk. So we had the data for 69 days, 30,000 people in total. So we had a big problem, like how are we going to visualize this data? what are we going to do with it? How are we going to look at it? So, of course, we have explored many options, and one of the particular things that we have explored is what is now going to follow. So we had the idea of, well, we started off with the idea, can we compare the different days? So we showed like all the nodes of the people that entered during one day, the connections are the very strong physical proximity between those people during those days. Just simply plotted in a circle with the standard layout algorithms. Then we showed the diagram. So the diagram is the longest, shortest path between two nodes, which gives you an idea of the size of the largest connected components. But there was time involved, of course. We had people coming in the morning who stayed 1 hour, 2 hours. We had people coming in the evening. Those people did not interact, obviously, because they were not present at the same time. So we introduced the notion of time through this scaled color grade, and we applied it to the nodes. So when a node entered, so when a person entered the room, the corresponding node was colored with the corresponding color according to the circle, which is plotted according to a clock. And then we additionally plotted, the amount of activity. Again, between noon and 1 o'clock, we had that amount of activity for slices of 5 minutes. So this is now a representation of the data for one day. Then, of course, we have this for every day. And this is an example, this is 4 examples of 4 different days. And the you see interesting patterns emerge. For example, at the bottom, you see like almost all nodes are connected, meaning that if there was an infection in the morning, almost everybody else could get infected by the evening. Whereas at the top, there is like different clusters. So there, this possibility, this potential vector is not present. Now, at the right, you see like 2 examples with the same nodes, approximately the same amount of nodes, yet with a completely different structure. For the first one is clustered, the other one is like connected. So at the bottom there might be a potential for having an infection going on during the day. At the top, it's simply impossible. And this is the kind of stuff that the real scientists are really interested in. Now, we put all this stuff together. So here we have Tuesdays, Wednesdays, Thursdays, Fridays, Saturdays in the columns. Mondays was the closing day of the science gallery. And we have 4 weeks going on. And then ended up with this big composition in which we have 69 days of clones encounters at the science gallery, which is the poster you have probably seen in the coffee room. So this is developed by myself, Wouter van den Broek, in collaboration with Marco Quagliotto, which is another designer, and then Lorenzo, Alain, and Ciro, who's present here, which are the actual scientists. I would also like to refer to the corresponding paper, which has been published in the Journal of Theoretical Biology. And I would like to thank the great organizations which made this research possible, in particular the SocialPatterns project, the Science Gallery, the ISI Foundation, and then CNRS, ENS, and Bitmanufactur. And just a little note, we also part in the studio on the project, so we're very happy about it as well. Thank you.
Thank you very much. The project, to lift the mystery, the StudiLab project is a project which brings together art, science, practitioners from various labs, and I think Rogers, home institutions also part of it. And I also like the idea to put up a poster, which is also a message to the humanities people, because I'm foraging both kind of areas, right? And what I see in conferences all the time, that in humanities conference, art history conferences, the poster session is always undergrad posters. You know, the lowly guys can do posters and the bigwig guys can do talks. But that's not true. A poster can be very powerful and reach much more people. And basically it's an awesome complement to this type of talk, because basically if you look at the 69 days on a slide versus at a poster, the poster is actually the better way to communicate it, which is awesome you brought one. So we have time for questions. Are there any questions?
on the highly affected infectious dates. What was the difference between the infectious dates and the others? Do you know anything about the audience? So, was this day what was so infectious? Was this only friends, or is there any kind of?
Honestly,
you don't know.
I think there might be a little bit misconception, because basically what we're showing was not. It was a potential for having a vector of infection going on during the day or being sustained during the day. And that, of course, depends on the time of arrival and the time of leaving. So when you have, like, what often happened during a regular school day is that you had a group of people coming in at, say, 10 in the morning, and they stayed for 1 hour until 11, and then they left, and then there was nobody in the room.
Okay.
And that's when you get, like, these, or you have, like, people, a group at the ground floor, and they happen to be in another room, and then you had another group at the top floor, and they left, and they did not meet really. Okay. It's more that kind of stuff. So it's not really highly infectious day or non-infectious day. It was a potential for having a sustained factor of infection that was highlighted in this poster, actually, or that was meant to be highlighted.
Is there a question behind?
So it seems like you spent a lot of effort in creating those beautiful visualizations and working through the data to be able to represent it in a way that you can extract meaning out of. So can you give us an example of the sort of high meaning that you got out of creating those visualizations? You know, what kind of insights did you gain into the behaviors, and how do you use that later on for, I don't know, I guess who you call the real researchers? You know, how do they use that now? Do you have any idea?
To be honest, no, I'm a designer. I'm a computer scientist, so I have more background. I deal more with creating the visualizations, and of course I communicate with the actual scientist. But it's a kind of question that would be more appropriate to ask for the scientist. Now, what can you actually get out of it? In this particular case, though, I know that this kind of visualization and the fact that they were all put together enabled them to understand this, or to better understand this notion of the presence of this vector of the potential vector, potential sustained vector of infection. So, and that you have this idea that it's not linear. So it's not like if you have that amount of people, you have this potential of having a vector. There is kind of a breaking point over which, like, of under which. So if you have a certain amount of people, and under that level, all of a sudden this vector is broken, and we don't have this diameter that spans the full network. So the network breaks apart in several components, which is quite interesting as a measure. So you could, like, extrapolate and say, like, okay, if we have a train station and we have a huge infection going on, what do we do? Do we close the train station, or do we just make that there's less people going in and out? So to break this sustained factor. And that's the kind of stuff, but you should ask them. There's sitting one here. He knows all about it.
Let's thank this. Oh, is there? Okay, there's one quick question.
Quick question. Yeah, you have wonderful data. I'm very impressed. And I want to ask if you try to develop some kind of epidemic model, like some differential equation, or like statistically analyze the data, like moving away from just networks. Yeah, maybe to you. Did you?
Yeah,
yes.
There are several. There is a BMC paper coming up in a couple of weeks about this.
Cool.
Look up the author's website, not only the speaker, and you will find a treasure trove of epidemics papers. So our last contributed speaker before we come to the discussion is George LeGrady, which is a notable artist about culture, data, and algorithmic organization. And I think let's let him speak for himself.